Hi, I'm Professor John Dalton. I lead a laboratory in molecular parasitology research at NUI Galway. Recently, we've been funded by Science Foundation Ireland to carry out research on COVID-19. As we know, COVID-19 now has become a pandemic throughout the world, and even in Ireland we've had greater than 60,000 cases so far. In collaboration with Professor John Doyle of NUI Maynooth, we are analysing the immune responses of individuals exposed to COVID-19. The purpose of those studies are to understand how individuals react to the virus and to determine whether they become immune and with time and what is that immunity associated with. For example, do they develop high levels of antibody, and are those antibodies capable of protecting them against the virus? Our first steps is to analyse the virus and look at its molecular structure and isolate certain molecules from the virus that we can use for diagnostic analysis. By understanding how individuals respond to certain molecules of the virus, we can then try to correlate those responses to protection or not, and we can also provide information be important for the development of vaccines. Developing diagnostic kits are not easy, but our laboratory are particularly expert at looking at infectious diseases, and in the next couple of videos my laboratory will describe the steps in which we're taking.
So after a detailed analysis of the genetic and structural makeup of SARS-CoV-2 virus, we selected a number of molecules that we considered the most suitable for developing our test. The genetic material that the virus uses to produce these molecules was then synthesized.
The synthetic genes that we produced will be then inserted inside the genome of a bacterium that will produce these proteins for us.
After that, we simply grow the bacteria in flasks like these, keeping them shaking and well aerated.
The bacteria make the protein for us. All we do is lyse the cells and isolate the protein using one of these.
We now check that the product is pure using the electrophoretic system. We're now ready to start testing.
The test involves using the purified viral molecule to determine if a patient has antibodies in their blood.
So as we can see here, the more antibodies a person has, the stronger the color reaction in the weight of this plate.
All the data is then analysed by computer to determine who has antibodies and who has not.
I hope that gave you an idea of what we're doing here in the NUIG to combat COVID-19.